Good evening, everyone. So, we'll be starting our class in 5 minutes so that people can join in. Okay? Yeah. We'll give 1 to 2 more minutes so that people can come and join in. Okay? In the meanwhile, I hope that you guys are up to date with the lectures which are being uploaded weekly. If you have any doubts or problems, feel free to ask in the class, or you can also reach out after the class as well. So, till now, we have almost say gotten the zest of the subject, and we got into some like details as well into the processes. So today we'll see the cell mediated immunity itself, like little bit about it, and then we're gonna solve some of the problems which are based on this week's lecture. So in this week, if you guys are up to date, then you know that certain tools and techniques were discussed, including immunodiffusion, ELISA, flow cytometry, right? Then blotting, electrophoresis, all these basic techniques which are oftenly used on daily basis in labs, especially where there is anything associated with protein or immunity itself. So there we use such immunohistocompatibility assays to make sense of the living tissue. Okay, so such things were discussed this week. And then and if time will permit, then we'll see some more questions. Okay, so just 1 more minute and then we'll start with this week's lecture. Okay. I guess we can start our class for today. So basically, in the last class, we discussed about the T cell receptors. If you remember, so we talked about monoclonal antibodies. Then we started talking about T cell receptors. Then how do they look? What is their basic structural configuration? How close they are with each other? What are protein families? Right? All this basic details we discussed. Then we also saw how basically the this CD4 and CD8, which are the T cell receptors, right, how they are selected in the system. Then we saw overall the summary of T cell receptor structure, how they function, what are their things which are there inside the cell. So receptor will definitely start a cascade of events inside the cell, right? So we just touched upon it little bit, and we'll see about it more in this, like in the current class. Okay, so if we see here, you can see we're going to discuss about the T cell mediated immunity, activation of T lymphocytes. So basically so there are few types of intracellular microbes, right? Intracellular means, so our humoral immunity was not able to combat the microbe or antigen fully, and they somehow evaded the blood, and then they are inside the cell. The antigen is inside the cell. Then how our body is going to take care of such pathogens which are inside the cell? So basically there are two such ways where the intracellular pathogen is taken care of. So if you look at the first, that is A, so what will happen? The macrophages are always searching for microbes, right? You remember, correct? So whenever there is a pathogen which is seen by the macrophage, what they'll do is they'll basically phagocytize it. Okay, so once they phagocytize it, so again it's a cell in which now the pathogen is there, right? So you can see here clearly, so this is the macrophage, and the pathogen or the antigen which was there has been engulfed or phagocytized. Okay, now imagine, now they are supposed to be digested because macrophages will digest them, but somehow what happened is they evaded the lysosome. So you remember, lysosomes are the chambers where the digestion of microbe will take place, right? But microbes are also smart, so what they did is they somehow managed to escape the this lysosome. Now if the phagocytic vesicle is combined with the lysosome, it is called phagolysosome, correct? So in the phagolysosome they managed to escape that. Now some examples which are commonly known and which causes the infection, which follows this trend of that they'll be phagocytized by the macrophages but they will escape the lysosome or the phagolysosome, they are mycobacteria such as Listeria, Legionella, certain fungi such as Cryptococcus, some protozoans as well such as Leishmania, Trypanosoma. So all these are parasites or microbes or antigens in this case, which are residing inside the cell. So if once we are saying that they are inside the cell, which means the antibody will not play any role in this directly. Okay, because antibodies will always be on the surface, but now the microbe is inside the cell, so antibody will not play direct role in controlling such infections. So what are the effectors which will assure that such infection is taken care of? They are CD4 and CD8. So by now you know that we are talking about cell mediated immunity, we are not talking about the humoral immunity. So in cell mediated immunity we have CD4 positive T cells and CD8 positive T cells. In CD4 positive T cells, they basically function by secreting cytokines and some associated receptors. Okay, so in the previous classes we have seen that only the recognition of CD4 or CD8 is not sufficient. We need co-receptors as well to be sure that this cell is the one which needs to be taken care of. Okay, so in case of CD4 positive T cell, they'll basically secrete, because remember CD4 positive T cells are also known as helper T cells. So they're gonna help in combating the microbe which is there inside the cell. Okay, and then in CD8 positive T cell, they basically kill the infected cell in which the microbes have escaped the phagolysosome in cytosol. And CD8 positive T cells, you know what they are called? They are called cytotoxic T cells, right? So they are going to directly kill the cell in which the microbe is present, and CD8 positive T cells gonna help in killing the cell in which the microbe is present. Okay, now we'll talk about the second pathway. The second pathway is basically all the cells which have been infected. So in this, a cell which has engulfed a microbe, and that microbe has somehow escaped the killing machinery of the cell, that cell has been targeted to CD4 and CD8. Okay, now imagine viruses, what they do is they directly infect the cell. There is no in between pathway for viruses, because they directly go to the cell receptor, inject their nucleic acid, and that's how they infect that particular cell. So what will happen is the microbe will be in a non-phagocytic vacuole, because they have not been phagocytized, they have been injected inside the host cell. So examples in which this is followed is obviously viruses. So most of the viruses work this way, like this is how they infect, and certain protozoas as well, such as Plasmodium, Cryptosporidium. So these are the protozoans which are not phagocytized by the phagocytic cells, but they are directly infecting the cells by their own machinery. Okay, so for such cells, which particular T cell will be activated? It will be cytotoxic T cell. Why? Because we need to kill this cell directly, so that no further infection can take place to the surrounding cells. So in such cases, CD8 positive T cells or the cytotoxic T cells will be deployed for handling such cases. Okay, now this is the overall induction and effector phase of cell mediated immunity. So till now, in the last, here we saw in what cases cell mediated immunity will help. Here we are seeing all the steps or major steps which are taking place in the cell mediated immunity. Okay, so imagine this okay, so this is a secondary lymphoid organ and a cell has come. So there can be two parts or two types of cells. First cell in which the microbe was there who has escaped the phagolysosome, and then another cell which has been directly infected by the virus. Okay, so in this case, in the secondary lymphoid organ, there will be B cells, T cells as well, but here the B cells are not responding anymore, because we are talking about intracellular pathogen. Correct. So here we will talk about CD4 positive T cells and CD8 positive T cells. All of them will be recognizing the cells by their surface receptors. So you know MH1 and MH2, whatever we have discussed, because T cells cannot see the pathogen directly, they have to see the pathogen only by MHC molecule. Okay, so one CD4 positive T cell recognizes the infected cell, one CD8 positive T cell recognizes the infected cell. What they'll do is they'll multiply, because this is the receptor which recognizes this, so they'll multiply so that more such cells can be deployed. So in both the cases such thing is happening, that is more proliferation and differentiation of the receptor which bound to the infected cell will be allowed to proliferate more. Okay, once they are proliferated, now they will be differentiated into the effector cells and they will be sent out to the circulation, so that they can reach to all those cells which are showing such receptors which are shown here, and they can then kill those cells. And here killing can be in two ways. first, if it is CD4 T cell, it will release some cytokines and then it will kill the cell, and by CD8 cell they'll directly kill the infected cell once they recognize it. Same thing, then they'll migrate to the place where the infection or this antigen has been is been present, and multiple cells are showing such receptors. So in the CD4 positive T cell, they'll basically, again the same thing which happened in the secondary lymphoid organ, it will happen at the peripheral tissue or the place where the T cells basically see the infection, and similar thing will happen. But here the proliferation will not take place. Here the mechanisms which are working for curbing the infection will take place. So if CD4 positive T cell identified the cell which had the intracellular pathogen or microbe, they'll release some cytokines, and what these cytokines will do, these cytokines will basically induce the inflammation, they'll enhance the phagocytosis, and ultimately will kill the microbe by killing the cell. So they are helping secreting these molecules which will lead to these things, and ultimately it will lead to the cell death which was infected. If it is the case of CD8 positive T cell, what will happen, this CD8 positive T cell will directly kill this cell. They don't need any cytokines, they don't need any secondary mechanisms such as inflammation or phagocytosis. They'll directly kill the cell which has been recognized by the differentiated CD8 positive T cell. Okay, this is the overall mechanism of cell mediated T cell immunity or T cell associated cell mediated immunity. Okay, now again this is a similar thing which we saw for the B cells, or how antibody or the B cell receptor or the secreted B cell receptor is selected, or how it is basically that type of cell which has the receptor is selected, allowed to differentiate and proliferate, and ultimately leads to the production of antibody. Similar thing happens in the T lymphocytes. So what will happen in T lymphocyte is that now in the secondary lymphoid organ, here there will be n number of CD8 positive T cells, CD4 positive T cells, but only one such cell will be able to bind to the infected cell. Okay, once this recognition happens, and APC here stands for antigen presenting cells. Okay, and antigen presenting cells are presenting the intracellular pathogen in this case. Okay, so once the antigen is inside the APC, the naive T cell, it can be the CD4 positive or it can be the CD8 positive T cell. Okay, once any one of it finds the better connection or recognition for this APC, they'll start to differentiate or say proliferate, because they make the connection. It gave this naive T cell the signal that, oh, I have matched up with this APC, and this APC is infected with a microbe, so I need more such cells which can bind to infected cells in this case. Then what will happen? A lot of cytokine secretion will occur, which will lead to the proliferation and differentiation of this type of T cell. Here it can be CD8 positive and CD8 positive. For both of them the process is same. Once this is done, then again they will be differentiated. So all this is happening in the lymphoid organ, secondary lymphoid organ, right? So once their number is good enough, they'll be deployed for the peripheral tissue where they'll do their process. So basically whatever we saw here, this is the in-depth info of the same process. Okay, so the effector T cell, so for example here if we are talking about CD4 or CD8 positive T cell, the effector cell, what they'll do is they'll do their designated function, and here the designated function is, if it is CD4, then it will release certain cytokines which will allow the infected cell to die via various processes, including inflammation. Complement part will also play a role here. Then enhancing the phagocytosis, all these things. But if it is a CD8 positive T cell, it will directly kill the target cell and hence completely eradicating the infection. Now we also need memory, right? This is the whole fundamental of how we attain immunity, how we get immunity as well, right? So once these cells, the selected cells, were proliferated and differentiated, the effector T cell will do the function. Some of such effector cells will be saved in the lymphoid tissue for future purposes, so that if the same infection comes again, there will be less time in handling the infection, because the cells will already be present, or in short, the memory will already be present in the system or in the host. Okay, so this is the overall process of how T cells or cell mediated immunity is taking place. Before today, we discussed about the humoral immunity, which also had part of CD4 positive T cells, CD8 positive T cells, antibodies, right? But here we are talking purely, completely about the cell mediated immunity, in which the secretion of antibody is not there. There is a direct contact with the help of receptors and co-receptors of the T cell and the antigen presenting cell with the help of MHC molecule, and then the infection is taken care of or is being eradicated or finished from the host. Okay. So these are basically you don't have to remember every single thing which is on this slide. Okay, so don't worry about that. So basically here what we are discussing is, we are discussing about the receptors which are present on the cell and the co-receptors or the ligand which is present on the other cell, so that the communication between the two cells, that oh, this is the infected cell, I have to take necessary steps to make sure that the infection is controlled, they are listed here. So if we talk about the receptors and signaling molecules of CD4 lymphocytes, so CD4s are basically what? Once they recognizes the cell, they'll secrete cytokines. Okay, but that recognition, how the CD4 positive T cell is recognizing an antigen presenting cell, is taking place. So here you can see it is not just the CD4 positive receptor or T cell receptor which is taking part here. T cell receptor is one receptor, but there are other receptors also responsible. So why do we need so many checkpoints? We can only have this T cell receptor recognition. Let it recognize the MHC second class second molecule, and let's start the process of secreting the cytokines and killing the cell which is showing this MHC2 molecule which has the microbe, right? Why do we need so much of co-receptors? Because imagine, all these receptors are what? Receptors are proteins. Okay, to make a protein, the cell basically needs to spend energy. The cell needs to undergo transcription, translation, then they are membrane proteins, so they have to be sent to the membrane in the right orientation, and in the meanwhile some proteins will not be proper, so they have to be degraded by the help of proteasomes. So all such process the cell is undertaking. Why? When only T cell receptor and MHC molecule will be suffice for this, right? Because this is the checkpoint, or these are the checkpoints to make sure at every instance that it is a self-host cell or it is a foreign cell. Okay, because all the microbes which are there, they are also made up of same things, right? Protein, lipids, carbohydrates, every single thing is same in the biological system. We all are carbon based life forms at the end of the day, correct? So to make sure that whatever I am recognizing, assuring all these checkpoints will help me be sure that the cell which I am going to kill now is actually infected, first thing, and this is the right choice for the killing. That is why we have so many co-receptors along with the main T cell receptor. So a signal transduction peptide is present, a receptor is present on the CD4, which is CD4, like the name itself says, because it has cluster of differentiation of CD4 type, 4 type, hence it is CD4. Then we have T cell receptor, then we have CD3, ITAM, CD28, CTLA4, inhibitory motif PD1, and then we have adhesion molecules as well, which help in recognizing the cell to cell interaction. Okay, and these are their corresponding complementary receptors on the antigen presenting cells. Okay, same thing goes here. So if we look here, the molecule on T lymphocyte is TCR or T cell receptor. They are present in all T cells because they are T cell receptors. CD3, they are again present in all T cells. Epsilon type, it is a basically signal transduction by TCR complex. CD4, they are present on antigen presenting cells. Okay, or they are expressed, basically their ligand is expressed on antigen presenting cells. CD8 positive, again their ligand is present on all the nucleated cells, and they are MHC1 molecules. CD28, same thing, their ligand will be on, or their corresponding receptor will be on the antigen presenting cell. So you don't have to remember all these things. Some major interactions you should know. For example, CD4 will bind to what? Like, okay, MHC1 or MHC2. Then what are their major common co-receptors, such as such as CD28, CTLA4 and PD1. Okay, so these are the major surface molecules on the T cell, and they are their receptors, or say their corresponding ligand on the antigen presenting cells. Okay, now again we are still in the cell mediated immunity part. Okay, so in the cell mediated immunity, when we talk about antigen recognition, now antigen has been recognized, we followed all the checkpoints. Now this T cell knows that this is the cell which has been infected with the intracellular pathogen. Now after this recognition, what will happen? This is the major recognition. This is the peptide or the microbe which has been shown to the T cell. Okay, so after that, downstream signaling needs to take place. Okay, so in downstream T signaling or downstream cell signaling, what happens is, once the APC shows the pathogen, this is the peptide of that pathogen, right? And the proper interactions have been made, then these interactions will lead to the downstream signaling, and depending upon the type of receptor they or signal they receive. So here if we are talking about CD4, which means this signal transduction will lead to the secretion of cytokines from this cell. Okay, if this was CD8, then this cell or this signal transduction will lead to the formation of cytotoxic commodities, which are ultimately going to kill the cell which the CD8 positive cell just recognized. Okay, so this signal transduction will depend upon what receptors have been activated. It is just like normal signaling. I don't know how many of you have been exposed to the cell signaling overall. So basically cell signaling, there are 3-4 steps in cell signaling. First is the recognition of the receptor with the ligand. Here the ligand we are saying is the processed peptide. This ligand can be anything. Imagine if it is a cytokine was also secreted. The cytokine will land on this cell. So once it is landing on this cell should have cytokine receptors, right? So at the end of the day, the receptor needs to have their ligand. Okay, so this is the first step. The first step is the receptor should be bound to the ligand. Second step, there will be some changes in the receptor. Okay, the third step, so here that change is basically phosphorylation. So this P stands for phosphorylation. When this complex was not made, the phosphorylation was not there. The moment this complex is made, there is changes in the receptor. In this case, it is phosphorylation. This is the second step. Okay, the third step is the secretion of secondary messengers. So this phosphorylation, so now these receptors which were not phosphorylated will not be recognized by proteins which are just present in the cell, but once they are phosphorylated, so their structure is changed, their chemical nature is changed. So some molecules which are there in the cytosol will now start recognizing this receptor. Okay, this is the third step. Once they start recognizing, they'll start activating the other protein molecules. That is the fourth step and basically the result. So here the signal transduction. How the signal is being transferred or transduced? The receptor has recognized its ligand. The receptor changed something in the cytosol, leading to the formation of secondary messengers, and ultimately the signal is generated. So these are the basic steps in signaling or cell signaling. Same thing is happening here, but here we are talking completely with respect to the CD4 or CD8 or any like T cell, how cell mediated immunity is working. Okay, now we'll move towards the signaling transduction which I just mentioned. There are some standard signalings which occur in the cell. The ultimate end step of the signaling is to change the biochemical nature of the cell. Let me summarize it. So here if we say to signal transduction, what possible things can happen via this signal transduction? So if it is CD4 positive T cell, which means this cell after making this complex needs to secrete cytokines. Okay, now how the cytokines will be secreted? Okay, someone has to tell this cell's nucleus that we need to make cytokine, and those cytokines need to be secreted. So what is going to tell this cell's nucleus that do something, make cytokine? This can be done via various signaling pathways. Okay, or various things which are changing the inherent nature of the cell which has just received the signal, and here by signal I mean a ligand. Okay, once we see here, so these are some major signaling pathways. They are not restricted to immunity, they are not restricted to CD4 or CD8 only. These are general signaling pathways which are present in the cell, but they can be stimulated or activated by different receptors. Here one of such receptors we are talking about is CD4 or CD8 or a T cell, because we are talking about T cell. Here if you take any other cell, for example you can take macrophage. Macrophage has cytokine receptor. Okay, so that cytokine receptor will ultimately activate any of these signaling pathways and many more, because these are not the only signaling pathways, and many more, to come up with the action or the thing which that cell is supposed to do. Okay, so here if we see a few such examples of signaling pathways, so this part is same. So if you can see before this purple block, 1 minute, this part, this is exactly same as this one. There is no change, no difference. Okay, now if we focus on this, we understood what happened. The complex is being made, the co-receptors have been activated, the change in the receptor has taken place. Now this is the first step in signaling, as I have said, right, that the ligand binding has taken place, or a complex is basically made. Now due to the phosphorylation of the protein which is mentioned here, protein, for example, if we take the name, it will be easier to understand. You don't have to worry about these names, okay. So ZAP70, so it is basically a protein only. Now this ZAP70 was just present in the cell, but it was not able to recognize this receptor which was already there, because it has the binding pocket for 2 phosphates which are linked to this receptor. Once this receptor got the phosphorylation, right, then this protein identified the phosphorylated protein or receptor. Once this protein is activated, it will have, here it is shown that it is phosphorylating another adapter protein. Once this adapter protein is activated, again this will be recognized by other protein which was present but was not able to recognize the adapter protein because it was not phosphorylated. Now once this PCL gamma is attached to the adapter protein, what it will do? It has functions. What function it has? It will increase the cytosolic calcium. It will release DAG or diacylglycerol. Once the cytosolic calcium is increased, this increased will lead to the activation of another enzyme which was not active, but once the calcium levels are more, this calcineurin enzyme is activated, which basically activates the transcription factor NFAT. This NFAT, because it's a transcription factor, so what does transcription factors do? Transcription factors basically go inside the nucleus, bind to the designated place or complementary place on the DNA, and leads to the transcription. Okay. Similar thing, DAG, if DAG is there, DAG will activate PKC pathway. PKC pathway, what it does? The transcription factor for PKC is NF kappa beta. Similar thing, another thing, so for example, more such proteins will identify the phosphorylated adapter protein. So GTP GDP exchange of RAS RAC pathway, so RAS GTP will activate ERK MERK pathway. Okay, activation of PI3 kinase will lead to the activation of AKT or mTOR pathway. So both of these, all of these enzymes which are mentioned here will ultimately lead to the change in the transcription factor profile, and once the transcription factor profile of a cell is changed, its transcription profile will change. It will make proteins for which the transcription factors are now available, and it will increase the protein synthesis. Once this protein synthesis is increased, so what sort of proteins are increased? They are increased which are regulated by NFAT, the proteins which are controlled by NF kappa beta, the proteins which are associated with AP1. So all these transcription factors will increase the protein synthesis of those proteins only which are regulated by such transcription factors. So can you see how just one ligand association or receptor binding to its ligand is changing the biochemistry or the protein profile of this cell? Okay. Again mentioning, don't be confused that these are the only pathways which a cell is taking place. These are general signaling pathways which are present in all the cells, but how they are activated is making the difference. Okay. So the overall summary for cell mediated adaptive immunity is: T lymphocytes are the mediators of the cell mediated arm of the adaptive immune response, which combats microbes that are ingested by phagocytes and live within these cells or microbes that infect host cells. T lymphocytes also mediate defense against some extracellular microbes, helps B lymphocytes to produce antibodies and destroy cancer cells. The response of T lymphocytes consists of sequential steps following recognition of host cell associated microbes by naive T cells, expansion of the antigen specific clones by proliferation, and differentiation of some of the progeny into effector cells and memory cells. T cells use their antigen receptors to recognize peptide antigens displayed by major histocompatibility complex molecules on antigen presenting cells, which accounts for the specificity of the ensuing response, and also recognize polymorphic residues of the MHC molecules, accounting for the MHC restriction of T cell response. Antigen recognition by the T cell receptor triggers signals that are delivered to the interior of the cell by molecules associated with TCR and by the co-receptors CD4 and CD8. The binding of T cells to APCs is enhanced by adhesion molecules, notably the integrins, whose affinity for their ligands is increased by antigen recognition by the TCR. APCs exposed to microbes or to cytokines produced as part of the innate immune reaction to microbes express co-stimulators that bind to receptors on T cells and deliver necessary second signals for T cell activation. The biochemical signals triggered by T cell by antigen recognition and co-stimulation result in the activation of various transcription factors that stimulate the expression of genes encoding cytokines, cytokine receptors, and other molecules involved in T cell responses. The TCR signaling pathways involve protein tyrosine kinases, which phosphorylate proteins that become docking sites for additional kinases and other signaling molecules. The signaling pathways include the calcineurin NFAT, RAS-MAP kinase, and PI3 kinase mTOR pathways. In response to antigen recognition and co-stimulation, T cells secrete cytokines that induce proliferation of the antigen stimulated T cells and mediate the effector functions of T cells. T cells proliferate following activation by antigen and co-stimulators, resulting in expansion of the antigen specific clones. The survival and proliferation of activated T cells are driven by the growth factor IL-2 or interleukin 2. Some of the T cells differentiate into effector cells that are responsible for eradicating infections. CD4 effector cells produce surface molecules, notably CD40L, and secrete various cytokines that activate other leukocytes to destroy microbes. Other activated T cells differentiate into memory cells, which survive even after the antigen is eliminated and are capable of rapid response to subsequent encounter with the antigen. Naive T cells migrate to peripheral lymphoid organs, mainly lymph nodes, draining sites of microbe entry, whereas many of the effector T cells generated in lymphoid organs are able to migrate to any site of infection. The pathways of migration of naive and effector T cells are controlled by adhesion molecules and chemokines. The migration of T cells is independent of antigen, but cells that recognize microbial antigen in tissue are retained in these sites. Okay. These are some of the characteristics of helper T cell, or say CD4 positive T cells. So CD4 positive, they are known as helper T cells. So they are helping in some function, right? They are not directly killing the microbe, correct? So how are they helping? So even in TH cells or T helper cell or CD4 T cells, there are few variations. So few effector T cells, for example, TH1, TH2, TH17, TFH. These are T helper cells only, but different versions of it. So if TH1 cell is there, it will be defined by cytokine interferon gamma or IFN gamma. The responding cell will be macrophages. So whatever we have seen in the previous slide, they were predominantly TH1. Okay. And how are they responding? They are basically helping the macrophage to be activated. How they'll be activated? Because they'll start receiving cytokines. Okay. And where TH1 is commonly shown more prominent results in is in autoimmunity and chronic inflammation. Similar thing if we talk about TH2, then the cytokines which are defining them, they are interleukin 4, interleukin 5, interleukin 13. Okay, they are majorly activated against helminthes or parasites. Okay, and in allergy. So the overall term or the umbrella term in which TH2 will be playing major role will be in allergy. TH17 on the other hand will secrete IL-17, IL-22. Okay, they are majorly for extracellular bacteria and fungi. They play major role in autoimmunity and inflammation. Similar thing for TFH. Here it will be IL-21, IL-4 and IL-13. They help in, or how like they are helping, so that function is antibody production. So wherever there is B cell taking help from the cell mediated immunity, CD4 positive T cell, the majority of that T helper cell will be of type TFH. Okay, and their main role in diseases where they play major role is autoimmunity or autoantibody production. Okay, so there is a very close knit control between all these helper T cells, right? Because if you remember when we were talking about antibodies, so one B cell will secrete one type of antibody, another B cell will secrete another type of antibody, but at one point of time you are not having only one infection, right? So if you have breached your skin or you have a rash or you got a wound, so all sort of infections can enter from that wound, right? Because the first barrier of immunity is broken. So anything which is there in the surrounding or in the environment can enter. So all these things, they'll be activated at the same time depending upon the sort of infection your body is trying to deal with first. Okay, so whatever and however we are studying, it is completely based upon to make things little bit easier. So imagine if you have a bacterial infection, right? It doesn't mean that you are not having any parasitic infection or you're not having any viral infections, all those sort of things. So everything is happening simultaneously. Okay, here we'll talk about the major responses of the cytokines which are secreted by helper T cells. Here we are talking about helper T cells in general. We are not talking about TH1, TH2, no. Here we saw with respect to the various types of helper T cell what they secrete. Okay, now we are seeing whatever they secrete, what is their action, like why are they being secreted. So it's a summary of that. Okay, so if you look at the first part, so these are general properties of T cell cytokines. So first, I hope this is visible to you guys. Let me zoom it a bit. I hope this makes sense now. So they are, the property produced transiently in response to antigen. What is the significance? It provides cytokines when needed. So immediate secretion of a cytokine. Usually act on same cell that produces the cytokine, that is autocrine type of signaling, or nearby signaling, also commonly called as paracrine signaling. What is its significance? Most effects are at tissue sites of infection and inflammation. Okay. Then pleiotropism, that is each cytokine has multiple biological actions. Pleiotropism means multiple things. Okay. It provides diversity of actions but may limit clinical utility of cytokines because of unwanted effects. Redundancy, that is multiple cytokines may share the same or similar biological activities. Therapeutic blocking of any one cytokine may not achieve a desired effect because there are multiple cytokines secreted simultaneously which have overlap or pleiotropic effects. Okay. Now if we talk about the biologic actions of T cells cytokines, or specifically T helper cell cytokines. Okay. So if we say IL-2, which was secreted by which cell? Do you remember? So if it then IL-2 was secreted by Th1 majorly, but because Th1 is the commonest one, right? This is what we have seen. So all the activated T cells will secrete IL-2. It's a universal interleukin which will be secreted. The action of IL-2 is, so IL-2 again, it's a small molecule, it's a ligand. So take this IL-2 as a ligand and paste it here. So imagine if this was not a T cell receptor but was a IL-2 receptor. Okay. So IL-2 receptor will receive the IL-2 molecule which is secreted by Th1. Okay. So the downstream signaling of IL-2 molecule is T cell proliferation, regulatory T cell development and maintenance. What is the source? So the source is obviously all the T cells, predominantly Th1, but all the T cells will be secreting IL-2. Okay. Then interferon gamma, activation of macrophages, that is the classical pathway. So T helper cells classical function is to help the B cell, okay, or help the cell to achieve its function because it is T helper cell. So the classical pathway will be activated by the interferon gamma. So what are the sources of these cytokines? It will be CD4 positive Th1 cell and CD8 positive T cells, natural killer cells, and ILC1. If we talk about IL-4, so B cells switching to IgE, alternative macrophage activation, CD4 positive Th2 cells and mast cells. Again I am reiterating that you guys don't have to worry about every single function or every single source. Just remember the major things of these tables. Okay. Then because at this stage you guys don't need such deeper information, I am covering all such things so that you have a better idea of when we say okay cytokine is released. So what is actually happening when we say that a cytokine is released from a T helper cell and it is eradicating the infection. So how it is eradicating the infection, how it is taking part in the immunity. So all these things I want to be clear so that you can have a better idea. Okay. Then we talk about IL-5. So the activation of eosinophils, CD4 positive Th2 cells, mast cells, and ILC2. IL-13 or interleukin 13. So B cell switching to IgE, alternative macrophage and activation. So here you can see IL-4 and IL-13, both of them has same function or same mode of action. So this is proving the property of redundancy, that two cytokines are performing the similar function. Okay. For IL-13 it will be CD4 positive, Th2 mast cells and ILC2. When we talk about IL-17, it is basically doing stimulation of acute inflammation. CD4 positive Th17 cells, ILC3 and other cells are their sources. For IL-21, B cell activation and TFH differentiation. Okay, so CD4 positive TFH cells. IL-22, that is maintenance of epithelial barrier function, CD4 positive Th17 cells, NK cells and ILC3. Okay, what are ILCs? Because it's multiple times it has been repeated here, right? So what are ILCs? I have only talked about TH, CD, T cell, B cell. What are ILCs? So ILCs are innate lymphoid cells. Okay, the cells which are already present in the lymphoid organs. Okay, apart from the B cell and T cells. Now this is again the similar thing which we have discussed during the start of this class, how CD4 and CD8 are basically killing the infected cell. So an APC which has phagocytized the microbe via its MHC molecule will display it to the CD cell, to the T cell. If it is CD4 cell, what it will do, because this is macrophage, so if you look here, interferon gamma, so the moment CD4 positive T cell is there, it will lead to the activation of macrophage. Okay, so what will happen, once the macrophage is being recognized by the CD4 positive T cell, the cytokine now which it will secrete is interferon gamma, leading to the activation of macrophage, and then the microbe will be killed due to, it can be with the help of phagolysosome, inflammation or any other thing. But once it is being recognized, once it is killed, once it is recognized by CD8 positive T cell, because the killed microbe is now being displayed here, the CD8 positive T cell will directly kill the microbe by secreting porins or multiple things which will lead to the cell death on spot. So this is how CD4 and CD8 are working in conjunction with each other to reduce, control and eradicate the infection. Okay, so this is the theory part for today. Now we're gonna switch gears a little bit to the tools and techniques which was discussed in week 6. Okay, so how we are going to discuss it, because majority of the classes of this week are pretty elaborative, they are pretty good enough in themselves, so I'm not covering them, but we'll definitely see some related associated videos to give it the visual impact of an experiment, so that you can see how the for example how the diffusion assay looks like, or how the ELISA works, so what is flow cytometry. So all those things we'll cover with the help of visual aids. Okay, so before starting, you know the drill. For easier questions, you will get 1 minute to answer. You can give your answers in the chat box. And second, for difficult questions, depending upon the level of difficulty, we'll either go from 2 minutes to 5 minutes. Okay, so for today, our first question is, let me get a laser pointer. So the first question is, in radial immunodiffusion assay, the area of precipitin ring is proportional to concentration of an antigen, concentration of an antibody, concentration of both antigen and antibody, none of the above. Okay, It's an easy question, so we're gonna have 1 minute for this, and your time starts now.
Concentration of an antigen.
Can you please drop your answers in the chat box? I understand that you want to answer, but it will be better if everyone has equal time and all. Okay, so anyways, after 1 minute we're gonna discuss this. Your 1 minute is about to be over, and the right answer is concentration of an antigen. You were correct. Let me see how many people have actually tried to give an answer. Oh, I started from a different ID. Okay, it's okay. Yeah, so we'll basically see. I don't know how many of you actually did the immunodiffusion assay, but it's really nice to see those inhibition zones and everything. Okay, so just give me a minute.
In the previous video lecture, we covered the concept of precipitation reactions in diagnostic immunology. These precipitation reactions are the basis of many techniques in immunology. Let's say, this is a test tube containing antibody molecules in a solution. Now we add soluble, non-particulate antigens in this test tube. After some time, these antigens and antibodies diffuse towards each other. When they reach their optimal concentration relative to each other, precipitation reaction takes place. This region is known as zone of equivalence. Recall that no precipitation occurs in the zone of antigen or antibody excess. The insoluble immune complex, formed at the zone of equivalence, is visible as a precipitation ring at the interface of two solutions. This test, illustrating the precipitation reaction in solution, is known as ring or interfacial test. Now here, the precipitation reaction occurred in a solution. Precipitation reactions can also take place in gel medium. One such technique in immunology is known as immunodiffusion. Immunodiffusion method is based on the principle of precipitation reactions between antigens and antibodies. Let's understand the basic technique of immunodiffusion. Let's say, this is an agar plate. Two cylindrical holes or wells are made into this agar. Now we had a solution containing antigen molecules in this well at the top. And in this bottom well, we add solution containing antibody molecules specific to the antigens. After some time, antigen and antibody molecules diffuse towards each other through this agar medium. When they reach their optimal concentrations, that is at zone of equivalence, the insoluble immune complexes are formed which are visible as a distinct Bander line of precipitation. This technique of immunodiffusion is mainly used for determination of the relative concentrations of antibodies and antigens, comparison of antigens and Determination of the relative purity of an antigen preparation. There are two important immunodiffusion techniques. First is known as radial immunodiffusion. Second technique is known as double immunodiffusion. In this video lecture we will cover radial immunodiffusion. Radial immunodiffusion is abbreviated as RID. It is also known as Mancini technique. This technique is mainly used to determine quantity or concentration of antigens, antibodies, complement proteins, etc. Sometimes, it is also called as single radial immunodiffusion. This is because, in this technique out of the antigen and antibody, only one is free to move. Suppose, we have an antigen solution of unknown concentration. Let's designate it by letter X. Our aim is to determine its concentration. This is a solution containing antibody molecules against this antigen. Also, we have 3 standard antigen solutions. Now, what is standard antigen solution here? This refers to the antigen solution of known concentration. Let's say they have concentration in increasing order. First contains the minimum antigen concentration, and third contains the maximum antigen concentration. Firstly, solution containing antibody molecules is added to the molten agar, and this agar is poured on a glass slide. Once agar is set, wells are cut out in the agar. Generally, 4 to 8 wells are made in the agar slide. For our illustration, we will take 4 wells. In first 3 wells, standard antigen solution is added. Let's label these wells according to standard solution number as 1, 2, and 3. Recall that standard antigen 3 has maximum concentration. In the 4th well, we add antigen solution of unknown concentration. This slide is now left for 24 to 72 hours. During this time period, the antigens diffuse out of the wells. This diffusion occurs in all directions. Now we know that antibody molecules are uniformly distributed in the solidified agar medium. So, when these antigens reach their optimal concentration relative to these antibody molecules, they form insoluble immune complexes. This precipitation reaction is visible as precipitation rings surrounding the wells. Now here you can see that size of ring varies. The size and diameter of this ring is related to the concentration of the antigens. More the antigen concentration, wider the ring. So, here you can see that precipitin ring surrounding the well labeled 3 is largest among all. This is because the antigen concentration in this well is maximum. The quantity of the antigen X is determined by standard curve.
It was a 1 hour disconnection. Okay, so don't worry. Moving to the next question for today. Yeah, I hope the slides are visible to you guys. So, and the second question for today is, an exogenous protein was overexpressed by a student in some cell lines. Okay, which technique would be the best to determine the overexpression of this protein in the cell lysate? So here you need to understand the key term which we want to identify, because there are n number of assays or immunoassays, right? But all of them do one particular thing for which we do that assay. Okay, so remember that and try to answer this question. It's an easy question itself. So I'll give you guys 1 minute and your time starts now. Your 1 minute is over and the right answer is Western blot. How many of you gave the answer? Great. Yeah, so I'm assuming most of you guys have at least done it once in your college, Western blot, because it's a very common technique and almost everyone does it daily if they are associated with any sort of protein purification, or even if they just want, like, if you are associated with a protein in a wet lab, for sure you have done Western blot and chances are you do it daily. Okay, so here's a very basic explanation of Western blot for those who have not done it, and for those who have even done it, so it will give you a little bit more clarification on the principles of why we do such thing. Okay,
In working with proteins, one cannot get around the technique called protein immunoblot, or simply the Western blot. Western blotting is a method to confirm the presence of a specific target protein in a sample. Before the analytical technique Western blot comes into play, everything starts with an SDS page. Upon treatment with SDS, all proteins in a sample are denatured and covered by negative charges. The proteins have a similar mass to charge ratio and travel through the gel to the positively charged anode. This allows to separate proteins according to their molecular weight. After staining the polyacrylamide gel, protein bands become visible. Marker proteins as a reference help to determine the molecular weight of proteins in the sample. This band here, for example, contains proteins with a molecular weight of approximately 60 kilodalton. However, that will not reveal which proteins are present in this sample. It could be the protein of interest, which has a molecular weight in this range, but the band could have also been caused by other proteins. To confirm the presence of a specific protein, a Western blot has to be performed. The detection of the protein of interest is based on antibodies, designed to exclusively bind epitopes of the target protein. The Western blot setup starts with the gel, containing the separated proteins. This is placed on top of a membrane, often a nitrocellulose membrane. This membrane is capable to bind large amounts of protein. Later, the idea is to transfer the proteins from the inside of the gel onto the membrane. This gel membrane construct is flanked on both sides by filter papers. All parts here are soaked in buffer to facilitate the electricity transmission later. Finally, everything is fixed between the cathode and the anode. The electric current pulls the negatively charged proteins, which are located in the gel, to the positively charged anode. This transfers the proteins onto the membrane. From now on, the membrane has to be incubated several times. The first step includes blocking. Therefore, the membrane is incubated with milk or BSA. Blocking should prevent antibodies from unspecifically binding to the membrane or to other proteins. After blocking, the membrane can be incubated with the primary antibody. This antibody should be selected to only bind to the protein of interest. After incubation with this primary antibody, a washing step will wash away the unbound antibodies while not removing the antibodies that are bound to the protein of interest. The next step involves the incubation with a secondary antibody, which has to be specific for the primary antibody. After washing, only those secondary antibodies remain which are bound to the primary antibody. The detection of the protein of interest is the last step in Western blot analysis. For that purpose, the secondary antibodies are conjugated to an enzyme. For many Western blot assays, secondary antibodies are conjugated with horseradish peroxidase. The protein of interest is bound by a primary antibody. The secondary antibody binds to the primary antibody, and when the membrane is incubated in a substrate mixture, the enzyme converts the substrate, thereby producing a chemiluminescent signal that visualizes the protein band. The presence of the target protein in these samples has been confirmed. Make sure to subscribe to stay updated for new videos.
It was pretty basic, right? So as I've mentioned, most of you have done it, but this is such a beautiful technique to think about, right? So we are using the basic properties of chemical entities and electricity and trying to extrapolate that what are the things present in a mixed sample, right? So we do this these techniques daily, but we forget how creative, innovative and changing they were once they were first introduced, right? So moving to the next question for today, it is basically with respect to immunostaining. So these are all the techniques which have been discussed in this week, so week 6th lecture. Okay, so in the immunostaining technique, paraformaldehyde is used as a fixing agent, permeabilization agent, blocking agent or detection agent. It's an easy one, but still, as the protocol suggests, I'm gonna give you guys 1 minute for this. Okay, and your time starts now. Your 1 minute is over and the correct answer is fixing, obviously. I hope even if you guys have not done it, yeah, great. Even if you guys have not done it, if you are from such field, you might do it, but it's not as frequent as a Western blot. Okay, so we're gonna see what exactly
We provide forward-thinking leaders.
Immunostaining. Let the ads skip. Yeah.
In this chapter, we will focus on the very heavily used technique of immunostaining and chemical dyes. To give you a short overview, I will first explain why immunostaining is one of the most common techniques used in the biological laboratories. Then, I will introduce the principle that underlies immunofluorescence, explain how more than one structure can be analyzed from the same sample, using multiple channels, and what to consider in these circumstances. Immunofluorescence is often coupled with the application of chemical dyes, which I will also shortly talk about. In the end, I will provide you with a general protocol for immunostaining. After recovering an embryo, the information that can be obtained from a bright field image is very limited, as we cannot make out specific structures. Immunostaining provides a fast and much cheaper way to obtain information on the localization of a wide area of proteins than generation of a transgenic line would, as that takes a lot of time and requires a lot of resources. It also allows to stain multiple structures at the same time and gives a high resolution, allowing in this case to make out the nuclei in blue, the cell shape in red, and the extracellular matrix in green of the embryo that we see on our left side as a bright field image. So, how do we resolve the localization of a specific protein? We use an antibody that was raised in a host species against our protein of interest, so that it binds to that with a high specificity. This primary antibody is then detected by a secondary antibody, which itself is specific against the species that the primary antibody was raised in. The secondary antibody is tagged with a fluorophore or horseradish peroxidase, which allows us to detect signals specifically at the localization of the protein. To allow localization of several proteins at the same time, to perform colocalization analyses to understand whether specific proteins are in spatial proximity, which would allow active signaling, for example, or to understand the localization of two different transcription factors in combination with cell-cell junction staining, we need to stain multiple proteins at the same time. For this, we must use primary antibodies that are raised in different species. The species most commonly used are mouse, rat, rabbit, and goat. Given a protein complex of 4 different proteins, we would use one primary antibody from each species to detect each component. Each of these antibodies will then be detected by a different secondary antibody, which is tagged with the specific fluorophore, resulting in a 4-colored image. Often, immunofluorescence staining is carried out in combination with the incubation of chemical dyes. These are small molecules that are either fluorescent by themselves or peptides that can be linked to different fluorophores and detect various cellular structures with a high specificity. Most commonly used are stains for the nucleus, as these allow quantifications of cell numbers. DAPI is a fluorescent molecule that intercalates with the DNA. Another family of nuclear stains used in several labs are called Hoechst stains. These also intercalate with the DNA but are less toxic for the cells, so that then they can even be used for live cell imaging. To gain information on cell shape, we can take advantage of phalloidin. Phalloidin is a bicyclic peptide that binds specifically to F-actin, a member of the cytoskeleton. It is a toxin found in mushrooms and can be coupled to fluorescent dyes that allow detection. If using phalloidin, we must consider that it provides us with some information of the cell shape, but it is not equal to the cell shape, as it localizes predominantly apicolaterally, but much less at the basal surfaces. The apical surface here is towards the middle, and the basal surface towards the outside of the structure. To gain information on cell shape, we would require cell membrane staining. To perform immunofluorescence staining, we can use the following protocol. Following dissection of the embryo, it needs to be fixed in order to preserve cell structure and protein organization, otherwise the embryo becomes degraded. Different fixation methods exist. Usually you can find a working fixation method for a specific antibody in the method section of publications in which the antibody was used. Most commonly used are methanol and paraformaldehyde, short PFA. For the first, the embryo is pipetted into ice-cold 100% methanol and incubated for 10 minutes at -20 degrees. For PFA fixation, the embryo is incubated for 20 to 30 minutes at room temperature in 4% PFA dissolved in phosphate buffered silane, short PBS. Following fixation, the embryos should be transferred back to PBS, supplemented with 0.1% green 20, called PBST, and washed 3 times. To allow antibody penetration through the cell membrane, the embryo must be permeabilized. For this, the embryo is placed for 20 to 30 minutes in permeabilization buffer, which is composed of 0.3% Triton X100, 0.1 molar glycine in PBS at room temperature.
Don't get overwhelmed. So if you do this, you have to follow this protocol like from A to Z, but in general you know the zest of the immunostaining, why it is used, why we do that, and where exactly these agents are used in what step. Okay, so anyways these slides will be available to you. You can always go back to such videos and clear your concepts again. Okay, coming to the next question for today. So we have, what is the purpose of blocking in ELISA? Again, ELISA is another very common technique which is used in labs, and also it has diagnostic purposes as well. So one should know ELISA very well because it's very basic biochemical assay. Okay, so what is the purpose of blocking in ELISA? So there is a step in ELISA called blocking. So what is the purpose of that step? Is it to promote binding of antigens, to enhance the sensitivity of the assay, to inhibit the activity of enzymes, or to prevent the non-specific binding of antibodies? I'll give you 1 minute because it's again an easy question, and your time starts now. Your 1 minute is about to be over, and yeah, so the right answer is to prevent the non-specific bindings of antibodies. How many of you attempted? Yes, the full form of ELISA, to prevent great. So yeah, coming to the next question for today. Certain statements are made below regarding ELISA. So statement 1 is, it is a highly sensitive technique for detecting and quantifying soluble substances such as proteins, peptides, antibodies, and hormones. B, it uses antibodies labeled with enzymes that produce a measurable product when a substrate is added. C, the technique does not require separation of free and bound antigen. D, the color change in ELISA indicates presence of target analyte. E, it is possible to use monoclonal antibodies in ELISA. F, the intensity of color produced is directly proportional to the concentration of target analyte. So which of the following option contains combination of all correct statements? As it is a little bit lengthy question, I know most of you will get the right answer, but still I'll give you 2 minutes for this, and your time starts now. Yeah, I guess this is good enough time. So I'm hoping that you guys got the right answer. So the correct answer is D. So the statement C and E are incorrect. So the statement C says the technique does not require separation of free and bound antigen. We do need that, right? And E, it is possible to use monoclonal antibody, which is false because reasons. Anyone? ELISA employs enzyme-linked antibodies to produce a detectable signal. ELISA is known for its sensitivity and specificity in detecting various it's absolutely correct, but okay, great. So all of you gave the right answer, but I really want you guys to either know or explain it to me why we can't use monoclonal antibodies in ELISA. Okay, so have this answer with you. The next question for today is the correct sequence of Western blotting is performing the page, transferring proteins to the PVDF membrane. Proteins are denatured by SDS and by boiling. Incubate the PVDF membrane to primary antibodies and secondary antibodies subsequently. And the correct sequence will be, so you guys have 1 minute for this, okay? And your time starts now. Time is up, and the right sequence is proteins are denatured by SDS and boiling, then they are subjected to page, then they are transferred to the PVDF membrane, and last but not the least, in this case it will be the least, then they are incubated with the PVDF membrane and the antibodies are added, and then finally they are transferred to the PVDF membrane. So how many? Oh, great. You can cross check. Correct. I'm glad that all of you guys are participating in the quizzes and the question solving. So the next question for today is, in the immunostaining technique, the proteins are stained with antigen, Comasi brilliant blue, silver stain, zinc stain, antibody conjugated to fluorophore or radioactive probes. It's an easy question. We'll give it less than a minute, but still your time starts now. Time is about to be over. I know all of you have given the answer already, but for those who might be attempting it later, so we give this time. Okay, so the time is up, and the correct answer is antibody conjugated to fluorophore, right? So great. Specifically, great. Coming to the next question for today. So In flow cytometry, what does light emit as forward side scatter measure? Okay. Cell size, cell complexity, cell surface marker, fluorescence, or all of the above. Your 1 minute starts now.
One bench right here.
Your 1 minute is over, and the right answer is cell size. So FSC measures the cell size. Great. So we'll see what is, so it's a very brief video about flow cytometry, and obviously it's a very complex instrument at the end of the day, and you need some expertise to handle such an instrument. So I hope that most of you can have an experience with flow cytometer. It's a really cool instrument in my opinion.
Cell processes are controlled by complex and dynamic intracellular signaling networks. Surface receptors pass information into the cell by modifying cytoplasmic proteins. These modifications include phosphorylation, methylation, and acetylation, which control a broad range of cellular functions including growth, development, and apoptosis. Disruption of signaling networks often leads to disease. To understand signaling processes in normal and pathological conditions, identification and quantification of signaling proteins is necessary. Flow cytometry is ideal for such a task. Flow cytometry is a laser-based technology widely used to phenotype cells using antibodies directed against surface markers. It can also allow simultaneous analysis of multiple intracellular targets using antibodies that detect signaling proteins, their post-translational modifications, epigenetic modifications, and downstream transcription factors. The first step of intracellular flow cytometry is fixation of the cells using formaldehyde, which immobilizes proteins and preserves transient signaling events. Cells are then permeabilized with methanol or detergent, allowing antibodies to access to the intracellular space. Optimal fixation and permeabilization conditions depend on the location and biochemistry of the target epitopes and should be carefully considered to ensure successful antigen detection, especially when staining surface and intracellular proteins simultaneously. Since multiple signaling proteins and surface antigens can be identified using fluorescently distinct antibodies,
Can everyone please check your mics and mute yourself? Thank you.
flow cytometry can readily characterize signaling profiles at the single cell level within phenotypically distinct populations. This allows the characterization of the relationship between intracellular targets and enables detection of signaling events from rare cell populations within a heterogeneous sample. Multiplex analysis of these protein targets saves time and resources while producing data-rich results, making intracellular flow cytometry an invaluable tool to unravel the complexities of cell signaling.
Right, so it gives very beautiful results as well as very meaningful results. Our last second question for today is, in FACS, that is flow cytometry at the end of the day, cells are sorted by dilution plating until there are only single cell in each well of microtiter plate, the differential weight, electrostatic force or magnetic force. Time starts now. 1 minute. Time stops here, and the right answer is electrostatic force. Let's see. Great. Chandrama. That is serial dilution. Okay. Then in the immunodiffusion test, if two lines cross each other, then what it means? Two antigens are identical, two antigens are not identical, two antigens are partially identical, or none of the above. We just seen the immunodiffusion information, right? The visual aid. So what does, if two lines are crossing each other, what does that mean? Whether the two antigens are identical, two antigens are not identical, two antigens are partially identical, or none of the above. It's an easy question only, so I'll give the answer. The answer is that two antigens are not identical. You can refer to the video which we just saw for the immunodiffusion, and you will know why we are saying this. And I guess you might have. Great. Yeah, so you already know the answer for this. Okay. So this is all from my side for today. See you in the next class. So thank you so much for your patience. Bye bye.